everyone. We are the student of Shivaji Rao S. Junle College of Pharmacy, Asanga. Myself, Bhagyashree Jain, along with Pawan Jadhav, Trupti Jadhav, Ankit Jogi, studying in final year B Pharmacy. Our topic is pharmacological significance of oxazole moiety, guided by Dr. Sunana Gorgaonkar, Associate Professor. What is oxazole? Oxazole is an interesting heterocyclic organic compound which has major role in chemistry and industry. It constitutes an important building block for synthesis of variety of compounds due to its unique structure consisting of five-membered rings which contain one oxygen atom and one nitrogen atom. This aromatic compound is an essential component in the development of medical products, agrochemicals, materials, sciences, and organic synthesis due to its stability and reactivity. Properties. Physical property. It is a liquid. Its boiling point is 69.5 degrees Celsius. Pyridine-like odor. Miscible with water and some other organic solvent. Weakly basic. Dipole moment is 1.5 d. pKa value for acidic is 0.8. Molar mass 69.09 gram per mole and density is 1.050 centimeter cube. Chemical properties. First, aromaticity. Oxazole is a heterocyclic compound with an aromatic ring composed of carbon and nitrogen atoms. The stability of the molecule will be enhanced by this aromaticity. Basicity. The nitrogen atom in oxazole can act as a weak base and it can accept a proton to form a positively charged species. Reactivity. Oxazole can undergo various chemical reactions such as electrophilic aromatic substitution, nucleophilic addition, and reactions at the nitrogen atom. It may be alkylated, acylated, or undergoes other functional group transformations. Oxidation. In certain conditions, oxazole may be left exposed at an oxidation reaction and lead to various oxidative products. Hydrolysis. Oxazole may become hydrolyzed in the presence of strong acids or bases, which would result in breakup of its ring. Resonance. The high reactivity of the ring and its significant involvement in reactions involving both electrophilic as well as nucleophilic reagents correspond to the ionic resonance of the oxazole structure. Indeed, oxazole ring is capable of being cleaved to produce acids, amides, and imides during oxidation. So here the resonance of the structure is given below, where the bonds are shifted, and firstly the O gets the positive charge and nitrogen atom gets the negative charge, and further the resonance is seen. Reactions. Synthesis of oxazole. So first is Robinson-Gabriel synthesis. The Robinson-Gabriel synthesis is an organic reaction in which a 2-acyl amino ketone reacts and followed by dehydration to give oxazole. A cyclodehydrating agent is needed to catalyze this reaction. The reaction is given below. So here the reaction is given, where mechanism of the reaction is given, where due to protonation, the further reaction continues and gets the substituted oxazole. And dehydration in addition to that. The oxazole ring is less basic that the starting 2-acyl amido ketone, and so it may be readily neutralized. So, labeling studies show that most basic Lewis substance is amide oxygen and therefore it forms a part of oxazole. Second is reaction between acid amide and alpha hydroxyketones. Reaction between these gets tri-substituted oxazole. The reactions is given below. So this is the example where alpha hydroxyketosuccinate plus formamide due to dehydrogenation, diethyl dioxazole 4,5-dicarboxylate is formed, and due to ester hydrolysis and in the presence of barium hydroxide, it gives oxazole 4,5-dicarboxylate, and due to the decarboxylation, oxazole is formed. Third, Van Leusen reaction. Van Leusen's reaction is caused by the reaction of ketone with tosmic result in nitrile formation. Van Leusen reactions are especially effective in the manufacture of oxazole and imidazoles when they are used with aldehydes. So this reaction is occurred in the presence of tert butanol and potassium tert butoxide and gives cyanide compound. So, as per literature survey, we have different activities. First is antibacterial activity. Antibacterial activity are those agents which inhibits or stops the growth of bacteria. The antibacterial potential of a series of pyrazole derivatives of oxazole 5,1 moiety was synthesized by Argade ND et al. And evaluated against S. aureus, E. coli, and T. aeruginosa as reference medications for antibacterial activity. Ampicillin and streptomycin were utilized. Wang et al. Synthesized nitrosamine, which is used to identify bacterial resistance by determining if a bacteria produces beta-lactamase by preventing over-treatment and treatment errors. The identification of bacterial resistance can enhance treatment quality and efficiency while lessening patient suffering and treatment costs. 2,4-di-substituted oxazole were synthesized by Dabolkar et al., who then tested their antibacterial effectiveness against gram-positive bacteria like S. aureus and C. diphtheria, as well as gram-negative bacteria like E. coli. The typical medication utilized was ampicillin trihydrate, and inhibition zone was expressed in millimeters. Compound 28 showed strong efficacy against the different strains of bacteria. Tele et al. Synthesized a variety of oxazole 5 ions and employed ciprofloxacin and sulfacetamide as reference medication to test their antibacterial potential against a range of harmful microorganisms. The antifungal potential of the produced compounds against Candida albicans and Aspergillus niger was also investigated. The zone of inhibition was examined with clotrimazole and gentamicin. Compounds 34 and 35 demonstrated good antibacterial activity. Saloni Kakkar et al. Prepared 4 substituted aryl 2,4-di-substituted phenoxymethyl 4-oxazole 5,1 derivatives and used the couplet method to test their antibacterial potential against Xanthomonas citri and E. coli in comparison to the common medication streptomycin. Second is anti-inflammatory. Kumar G. et al. Synthesized anti-inflammatory properties for synthetic substances were tested against carrageenan. Three different dosages of 25, 50, and 100 mg per kg were used in the study. The anti-inflammatory activity of thiazole derivatives has been seen in to the range from 29.7% to 69.6%. At 25, 50, and 100 mg per kg compound, 3C had greater anti-inflammatory activity than the reference medication phenylbutanol, 100 mg per ml. The variety of oxazole derivative were prepared by Dunder et al., who then assessed their ability to inhibit COX-2. The constitutive form of COX-1 is involved in hemostasis and gastroprotective effects, while COX-2 is involved in inflammatory sites of compounds that were synthesized. It was discovered that 84 had the greatest selective COX inhibition. Shakya A.K. et al. Synthesized a series of N2,4-chlorobenzyl benzoxazole 5-YL3 substituted propanamide 3,8-2,3N and evaluated for their acute and chronic anti-inflammatory potential. These derivatives demonstrated excellent anti-inflammatory activity. Anti-cancer activity. These are those agents which cure activity. Chichua M.A. et al. Synthesized medications based on isoxazoles oxazole additionally reported are the corresponding hydrogenated derivatives or oxazolines or oxazolidines. These derivatives exhibited excellent anti-cancer activity as compared to standard drugs. Base oxazoles were synthesized by Cantalajo et al. And their anti-cancer activity was assessed against the cancer's line HT29. Additionally, the inhibitory potency of the derivatives towards recombinant human choline kinase CHOC K was evaluated in an ex vivo system. These derivatives were found to display potent anti-cancer activity. Johan et al. The synthesized special sequence that encloses the thiazole moiety in Aurora kinase inhibitors SNS 31424. Additionally, important binding components and SAR have been described. Using 5-fluorouracil as a reference, Louis et al. Prepared a variety of tri-substituted oxazole derivatives and evaluated their anti-tumor potential against two cancer cells, PC3, that is human prostate cancer, and A431, human epidermis carcinoma. This novel moieties exhibited excellent activities against the PC3 and A431 cell lines. Semen Utah et al. Synthesized oxazole derivatives based on the potential of anti-cancer activity. Now, anticonvulsant. So, effective synthesis of pharmacological potent 1,3,4-oxadiazole compounds was reported by Kamble and Suda, some of which contained triazoles. When compared to conventional phenytoin, it was shown that several variants containing triazoles exhibited very good effectiveness against convulsions generated by MES in rats. Su et al. Investigate a series of benzoxazole derivatives containing triazolone that were developed and synthesized as an extension of the previous study, substituting triazolone for the triazole compounds. Based on a theory that has previously been supported by earlier research, this design aims to enhance the anticonvulsant action of triazolone by increasing its affinity for the receptor. Guan LP et al. Synthesis of number of triazolone benzoxazole as anticonvulsant agents, among them 2-phenyl 6,4,5,1,2,4 triazole 4-benzoxazole was the most active and also had the lowest toxicity. The anti-MES potency test showed a median effective dose ED50 of 29.5 mg per kg, a median toxicity dose of 285 mg per kg, and a protective index of 9.7, which is greater than the reference drug carbamazepine, whose PI value was 6.4. These attempts suggested that the monosubstituted triazoles also meet the request of the anticonvulsant activity. Meanwhile, another series of triazole containing benzoxazoles were prepared by altering the position of the triazole. In this study, compound 20 was obtained with the ED50 of 12.7 mg per kg and 29.5 mg per kg in the MES and SCPTZ models, respectively. The Rotarod test showed the TT50 of 49.1, 491 mg per kg. Sidorenko I et al. Synthesized novel oxazole bearing 4-thiazolidones as potential anticonvulsant agents. It was found that this compound exhibited promising anticonvulsant activity as compared to standard drugs. Antidiabetics. These are those agents which are used to cure diabetes. So, Mariappan G et al. Synthesized and evaluated 4-arylidine 2,4-methoxyphenyl oxazole 5,1 derivatives of oxazole and found that 4,3-methoxybenzalidine 2,4-methoxyphenyl oxazole 5,1 was having activity equivalent to that of rosiglitazone. Ashton et al. Synthesized a range of beta-amino acyl piperidines with fused 5-membered heterocycles ring, that is thiazole, oxazole, isoxazole, or pyrazole. As dipeptidyl peptidase 4 inhibitors, out of all the screened oxazole derivatives, R3-amino 1, 2-cyclopropyl 6,7 dihydro oxazolopyridine 5,4-HYL 4,2,5-fluorophenyl butane 1 was found to possess considerable DPP-4 inhibition. A chain of oxazole derivatives was synthesized. Coulter R et al. Checked for PTP1B inhibitory activity. PTP1B has been found important for the treatment of diabetes and obesity. Out of all compounds, 97 and 98 exhibited the most promising activity. Pingali et al. Designed and synthesized 1,3-dioxone carboxylic acid derivatives and combined these with substituted oxazole and evaluated them for in vitro PPAR agonistic potential and in vivo sugar lowering and lipid lowering efficacy in animal models using rosiglitazone and tesaglitazar as standard compounds. Compound 99 was found to be the most active compound. Lastly, antifungal activity. These are those agents which affects or inhibits the fungal activity. Zhang et al. Produced a chain of several derivatives of propionic acid and used different reference medications to test their antibacterial and antifungal capabilities against different microorganisms. The antifungal potential of a series of pyrazoles connected to the oxazole 5-1 moiety was synthesized and evaluated by Candida albicans. Reference drugs for antifungal activity are fluconazole, ketoconazole, and clotrimazole were used for antifungal activity. Benzoxazole 5-carboxylate derivatives were prepared and their antifungal activity was evaluated by Shamsuzzaman Khan et al. Against fungal strains. Tom et al. Synthesized new derivatives of 5-membered heterocyclic compounds containing oxazole and benzothiazole rings and then screened them for antifungal activity using ketoconazole as a standard drugs. The novel oxazole derivatives were found to be the promising compounds for antifungal activity. Padmavati et al. Synthesized a new class of amide-linked bis heterocycles and tested their antifungal activity against coccidioidomycosis, histoplasmosis, blastomycosis, pneumocystis pneumonia using chloramphenicol and ketoconazole as standard medication. This new class of amide-linked bis heterocycles exhibited higher activity than standard antifungal compounds. So references. These are those references we have taken in our during the project. So the anti-cancer potential of oxazole has not been explored to a greater extent. Hence, we are making an effort to synthesize novel anti-cancer derivatives of oxazole. Acknowledgement. A special thanks to Dr. Shivaji Rao S. Jonle, Sir, as the President of our college, Sir Dr. Geeta Jonle, Ma'am, Secretary of our college, Dr. Nitin C. Mohire, Sir, Principal of our college. For more informative videos, please like, share, and subscribe to our YouTube channel. Thank you.